as we work with these microorganisms, they are unlocking new levels of bioavailability, fertility, and health in our soils. And we don't know what the limit of that is, quite frankly. Thank you, Aaron. Very glad to be here. Thank you, everybody, for coming in and participating to learn along. So, effective microorganisms and bokashi is something that is actually near and dear to my heart. And I mean that literally in the digestive system, and I also mean like I have an affinity for it. Now, the reason for this, I didn't actually find out about EM. Let me say some grammar here, actually. EM is a colloquial term meaning effective microorganisms. It was branded back in the 80s by a professor, Taro Higa. I do have his book, but it's in my rig. I don't have it with me here, shame on me. But I will quote out of it a few times as we go through this. And here in America, I do use a product by SCD, ProBio Balance Plus. And when I say EM, I am also meaning this. EM is the colloquial term, and it's the most commonly one. It is the one that is used on a global basis. So with that out of the way, what exactly is it? What's the hype with it? If you've gone into regenerative agriculture or tried your own gardening or looked into this, you may have run across EM and bokashi. Perhaps people were saying it's really good for your soil, it's really good for your plants, really good for your compost. Perhaps people were saying because it's anaerobic, it's bad anaerobes. And there's just kind of this mystery around it that has been there for a lot of years. But what we've been able to see is that anybody that has worked with it, cultured it, redistributed it, or partnered with it to any significant degree knows that it works, and it works pretty universally. And now, why is that? Well, let's start with what is it? It is a consortium of beneficial and active microbacteria. Consortium being a set of cultures that were cultured together and work in a symbiotic manner. So as far as EM in particular, we have a few MVP players. These are your lactic acid bacteria. These are your brewer's yeast. So right there, we're in familiar territory. These are what we use to make wines. These are what we use to make bread. This is what we use to brew beer. And these are very common when we ferment our vegetables or pickle something. We're partnering with a lot of lactic acid bacteria. Now, some of the not so common ones are purple non-sulfur bacteria. And this is a really fascinating bacteria because it is able to function aerobically with oxygen, anaerobically without oxygen. It is able to break down sugars, it's able to break down carbohydrates, or it's able to feed on carbon dioxide. It is also able to photosynthesize. Yes, photosynthesizing bacteria, like plants and trees, except instead of having oxygen as a byproduct, it just has a hydrogen as a byproduct. So it gives a proton. It's a photosynthesis that allows extra chemical energy to enter into the soil. So when you are culturing EM, as you're consuming all of the molasses, you will be releasing carbon dioxide as it gases off. Now, it's very common just to burp it, let that carbon dioxide out, but some people, or if you wanted to put a balloon on it, the purple non-sulfur bacteria will actually switch from carbohydrate mode to carbon dioxide mode and reconsume that. It takes time to switch between food sources. But it is worth knowing that it's in there. You also have rhizobacteria. These are bacteria that live in and around the roots of plants. So plants, when they start photosynthesizing, they have a bit of a shotgun effect. They just start putting sugars out into the roots, into the soil, and they're not specific as to what bacteria or fungi they're targeting. They're just like, hey, here's a food source, who's hungry? And then as more bacteria and fungi come into that rhizosphere, then they start having more selective relationships with these microorganisms. Acinetobacter. So this is very powerful stuff for bioremediation, for dealing with toxins, for dealing with chemical spills. It can't do everything. It's not a magic bullet, one-shot solution. EM stops right about where fungi steps in. So it can't break down tree lignin, only fungi can do that. But just shy of that, complex carbohydrates, proteins, animal meats, dairy, bones, all of this will ferment with effective microorganisms. We also have nitrogen-fixing bacteria. There's over 70 different types of bacteria that are in an EM cultured brew. So I'm obviously not going to go into all of them. I do not have them memorized. I know some of the key players, and I really want to share that I am a learner in this, and I have been learning and been fascinated by effective microorganisms for about 5 or 6 years now. And I've brewed many hundreds of gallons and done a lot of bokashi, redistributed and sold myself for a while. So I am not an expert, but I am an evangelist, let's say. Okay, that is all very exciting, very exciting. And Streptomyces. We forgot about Streptomyces. They are very effective at breaking down complex carbohydrates, and they are an immune response. So now that we have hopefully somewhat of a general idea of what a consortium is, what each of the individual pieces have, what is their cumulative effect? What is the effect of the consortium itself when applied? Well, the word bokashi actually is a Japanese word, and it translates directly to fermented organic matter, which is really fascinating. So fermentation is a process of microbes converting basic sugars, microbes and yeast converting basic sugars into alcohols, carbon dioxides, amino acids, humic acids. They will effectively chemically change organic matter into a bioavailable form. So let's take a look at my bokashi bucket in order to give us a better idea of what we are working with here. So let me see if I can zoom in on this for you all. Yeah, there is a bucket of food scrap. Yum. Now what you don't see is how it smells, but you can see what it looks like. And you see all the white fuzz that's growing all over it? That will be your Streptomyces. That's the stuff that's breaking down all of the complex carbohydrates. So that is where all of this really comes into play, is because with the consortium of bacteria, we are able to break down a wide variety of food sources. And as I said, I have 2 steaks in here, entire sunflower stalks. I even took some of the pathogenic culturing black green mold out of the garden and put it in there as well, because I'm that confident that EM is able to address these, because it operates at a pH of below 3.8 in an anaerobic condition. So not a lot of things survive that. You see I have an airtight lid on here. So all of your pathogens, all of your pathogenic fungi, your molds, it just doesn't survive. And as long as your culture is successful, you're not going to get any of that. Now, bokashi doesn't take care of weed seeds, and that is really unfortunate. But you also have this tap at the bottom, because you get a leachate that comes out of it, and the leachate is just a liquid runoff from what was being used with it. Now, where would you use this and why? Obviously composting, fermentation of organic matter in order to rapidly turn it into a bioavailable form for plants and as an inoculant itself, but in the roots, in your water, as you're watering, because these bacteria in a composting scenario ferment organic matter. In a soil scenario, they actually regulate the photosynthesis of the plant, and they regulate the sugar production of the plant, and they regulate the immune responses of the plant. All the while that they are cultured in the root zone, ensuring that no other pathogenic organisms live in the same environment. So this is where we get into the fact that they are facultative microbes. And for a long time we thought microbes were either anaerobic or aerobic, without oxygen or with oxygen. But when we think about more of the southern hemisphere in the rainforests, you have mostly flooded but occasionally dry soils. So these bacteria that are able to expand and survive in both conditions and to play different roles in each condition are what we would consider facultative. So when they're in composting, they have one role. When they're in the rhizosphere, they have another role. When they are sprayed as a foliar spray, they have an entirely different role since it's mostly aerobic. When we digest them, they have a different role again. Like purple non-sulfur bacteria, for example, it's in compost piles, it's in manure piles, it's in legumes that cows eat. And when the cows eat these legumes, the bacteria, they don't stop. They're now in the ruminant of the cow's digestive system, and they switch their roles from aerobic to anaerobic, and they play a digestive role in the cow's digestive system. So this is something that is vital to understand about microorganisms, is that they play different roles in different environments, depending on what is needed for them. And that's what's so fascinating about EM, is it has all of the microbes to do most of the things in most environments. And when they're done with their job, they bow out. They don't stay, they don't overculture, they don't overtake whatever it is they're growing into. When they've done their job and they've cleaned up the environment, they'll go dormant. They'll become food for the rest of the soil food web themselves. Some will stay, but nature has a very good way of keeping balance in things, especially when things become healthy. So that is why I rely on effective microorganisms to control pathogens, to keep my compost clean and free of disease, to rapidly process food waste, to inoculate the piles, to make sure that I have adequate nitrogen fixing bacteria, make sure that my plants are fully photosynthesizing. This keeps pests away. John Kemp's work has demonstrated that plants that are unhealthy leach nitrates, and they're entirely made up of simple sugars. Now your grasshoppers, aphids, mealyworms, all of these things that we consider pests in the gardens, they can only feed on nitrates and simple sugars, oxidized plants, because they only have simple digestion systems. But according to John Kemp's work, and from what I've seen with other people that have grown using these methods, if you have the right minerals, if your plant is fully photosynthesizing, and you have the right bacteria to maintain the rhizosphere, the plant is healthy enough that it no longer produces simple sugars. It actually builds itself out of complex carbohydrates. And it synthesizes all of its nitrogen in a 24-hour cycle. So all of these bugs that are looking across the infrared spectrum for oxidized and nitrate-rich plants, healthy plants are invisible to them because their digestive system cannot digest carbohydrates. Isn't that fascinating? We make our plants healthy enough and the bugs go away. Wow. Beyond that, with enough robust biology, plants actually produce lipids, fats, and then they store those in the soil as soil organic matter, which continues buffering everything else that happens in the soil. So we have a lot of reasons why we want our plants to be healthy. And what we find through this digestive process, through this breakdown, growth, regulatory process, is EM is a bioinformational feedback loop. It is a primary decomposer. It watches over the growth process. It watches over the immune process. And it starts again at the decomposition. When we start using it in our plants, we start producing healthier fruits and foods. And these are now inoculated with healthier bacteria, which go into our digestions, which in turn leads to healthier humans. It's a bioinformational feedback loop. So this is really important stuff. Okay, I hope I didn't ramble too much there, and we have the idea. Let's talk about working with it. What do you get when you get EM? So you have a mother culture. This is great. Shame on me because it's out in direct sunlight for this film. It doesn't like direct sunlight. It likes it being very dark and cool, but for the purposes of demonstrations, this is what we're working with. Let's go ahead and run our own here. And I will turn the lighting down just a little bit so that we can see what's going on. So EM is 1 part EM to 1 part molasses to 20 parts water. So I've already done the math. That is 0.8 cups of EM and 0.8 cups of molasses per 1 gallon of water. 1 gallon containers. Let's crack it open. Now, if you wanted to do 3/4 of a cup, you can do that. If you wanted to do 1 cup, you can do that. If you have done it as much as I've done it, you can just kind of do it by hand and watch how much the bottle goes up. And that is totally appropriate as well. So there I will add 1 part of the mother culture, 1 part of the unsulfured blackstrap molasses. Does anybody else have EM? Are they doing this right now? Nice. It is a lot of the same process. As far as that comment that I made, it was really just the distinction of if you are collecting on a starch water source, you're getting the lactic acid bacteria, but how do you know in your garden that you're also getting the purple non-sulfur bacteria and the streptomyces and the actinomyces and the rhizobacter if they're not already present? How are you going to get those in the first place? So it's similar, but it's not a complete consortium, if that makes sense. And then kind of just shake it gently like it's in the ocean. And there you go. That's it. It's really that simple. 1 part EM, 1 part molasses, 20 parts water. Store it warm if you can. In the greenhouse if you can. Under a towel. It likes it dark. The alternative to this is keeping it in your oven with the oven light on in a towel. And that will keep it warm enough to do the job as well. So that happens, and a few days later your bottle starts to look like this, and it's got a ton of pressure in it. That's that carbon dioxide that I was talking about that happens there. And let's see if I can get the microphone close enough that you can hear it. and then it vents off all that carbon dioxide. It goes back to there. You reclose it, let it sit for longer. Now, how do you know when it's done? Well, for one, if it smells sweet, you still have molasses. If you smell the mother culture, it's much more, it has a biological punch to it. It'll catch you back a little bit if you're not expecting it. So it shouldn't be sweet. You shouldn't smell the molasses because a fully activated version has already consumed all of the molasses. The other method is with a paper test strip. Anytime it goes below pH 3.8, ready to use. Now, once your ferment is done and you're ready to use it, it's good. Its maximum potency lasts for about a month. And you dilute that down 1 to 1,000 to use it in your water system. You dilute that down 1 to 1,000 to use it as a foliar spray. And you dilute it down 1 to about 2,500 to use for flowers, because it'll actually ferment flower petals if you use too much of it. Now, what you can also do is go to your local store and get either wheat bran, which is what I have here, or quick oats, rye, barley, any of these grains that will be able to be cultured onto. And then you take it into a consistency where you can just kind of squeeze it and water comes out, but when you let go, it more or less holds its shape. That's what you're looking for. Get that in an airtight container, let that sit for about 2 weeks. I started this one a couple weeks ago, and I was hoping we could see some of the cultures growing on it, but I don't see them yet. So we already did get to see those in the bokashi bucket, so that's okay there. But when you make the dried form of bokashi, once it's done, either put it in a dehumidifier or out on a warm day under a shade cloth to let that dry, and that's usable for about a year. And that's the most common method for the bokashi bins, is the dried powder form, so that every inch layer of food waste, you just sprinkle that on there. So now, what about human digestion? I do drink the EM that I activate, but they also do make them specifically for human consumption. So I would recommend going down that route. Okay. Wow. We covered a lot. How are you guys feeling? Did I ramble on too much?